Good afternoon, everybody. Welcome to the College of Engineering Virtual Brown Bag Series. We have been having these very interesting talks for the entire semester now, and we intend to continue this for a while, so welcome again. My name is Gautam Das. I'm the Associate Dean for Research in the College of Engineering, and it's my great pleasure today to invite to host the meeting and to invite Dr. Justyn Jaworski, who is a professor in the Bioengineering department, to give the talk today, the presentation today. So brief bio of Dr. Jaworski. He's an assistant professor in the Department of Bioengineering at UTA, and he's directing a research group that works on very interesting stuff, and we'll hear about some of that today, of using synthetic biology and soft matter bioengineering for the development of biomaterials and sensing technologies for disease detection. And he's co-authored over 60 publications, and he's got lots of funding from various agencies, domestic and international, including NIH, US Department of Defense, and National Science Foundation, National Research Foundation of South Korea. Interestingly, before joining UT in 2017, he was an assistant professor at Hanyang University in Seoul, South Korea. And prior to that, he received his postdoctoral training with a Nobel laureate, Sir Gregory Winters, at the MRC Laboratory of Molecular Biology in the UK. And he did his PhD from UC Berkeley. He is deeply committed to enhancing the undergraduate research experience, and the student projects have resulted in exciting outcomes, including the first 3D nanoscale protein printer. So with that, I would like to welcome Dr. Jaworski, but before he starts his presentation, just wanted to tell the audience that we hope to make this an interactive session, so I'm sure you'll have questions that you would like to ask. So there is a Q&A chat window on the screen. Please type your questions there, and whenever I get a chance, once Justyn has finished his presentation, I will read them out to him. So I hope we can keep this as informal and as interactive as possible. Justyn, over to you.
Thank you very much, Gautam, for the nice introduction. It's my pleasure to present to you all today about the research going on in our laboratory. So probably you guys know what sensors are, but just in case you don't, sensors are basically something that provides a measurable output for some type of event, usually a binding event, and that's something that you're interested in trying to detect. So those could be referred to as sensing systems, detection systems, tests, diagnostics. If you're at a hospital, you probably heard them called scans, depending on which modality people are using, or even assays. So sensors can be important for trying to identify all sorts of things. We could be looking to try to identify explosives if you're walking through the long lines at the TSA screening site at the airport, or something as simple as a glucose meter, right, for detecting blood sugar levels, or pregnancy tests, things like that we're more familiar with. So why are these biosensors really important? Oops. Really because it's the earlier detection you have for certain diseases, the better you are at being able to treat those diseases. Essentially, if you've watched the news or basically been aware of anything that's been happening in the past year or so, you're aware that people are trying to identify all sorts of ways in which we can better detect coronavirus exposure, right? Whether or not we have antibodies present for that exposure or that infection to coronavirus. And those can be kind of fairly invasive techniques, or uncomfortable techniques, we should say. But there's other types of biosensors that you're more familiar with, like pregnancy tests. And these are more or less very straightforward tests where you just see a visual output of like a yes or a no. There's other cases where biosensors are important where you have unknowns. So you may have some particular biotoxin that you might be have identified in somebody has gotten poisoned, and you want to identify what type of biotoxin you've been poisoned by, or there might be some virus that you previously didn't know existed, and now you're trying to identify what kind of virus this is. Or you might be trying to identify a type of disease that's what type of disease that you have. So there's all sorts of ways in which biosensors can effectively provide for a better quality of life by letting you know what it is you're exposed to or what it is you might have. So the key idea with biosensors is basically you have two elements in a very simplistic way. You have a transducer and you have some type of selective recognition element if you want to make a biosensor. So let's pretend in this example we're trying to make a sensor that detects the presence of a mouse. So that's pretty straightforward. We know mice like cheese. So could we just put a piece of cheese and connect it to an alarm? Whenever the mouse bites the cheese, the alarm goes off. So really, for a good sensor, you need to have something that connects that selective recognition event, that would be the mouse biting the cheese, into some detectable signal that we can all recognize. That could be an electrical signal, that could be an auditory signal, that could be a visual signal. And so let's just pretend you wanted to make a sensor for a mouse, right? You wanted to see if a mouse was present. Could you use cheese? I mean, I like cheese. I could very well fall victim to biting that cheese as well, and it would detect that I was present. Or maybe another animal walks by and bites the cheese. And so it's not a very selective sensor then, is it? It's giving you false positive signals in that another entity could be interacting with that recognition component. So you need to have that selectivity. And usually the sensitivity is represented by the transducer. How much interaction does the mouse need to have with the cheese before that alarm goes off? So for instance, what if the cheese jiggled slightly because there was an earthquake or a large truck drove by or something like that? Would that elicit a signal from that event? If so, you would have a false positive signal. What if some other animal, for instance, had tried to eat the cheese? We could put a box over this little sensor and have a small hole only big enough for a mouse to enter, for instance. So you can imagine clever little ways that you could try to make this fake sensor into something that's more selective and something that's more sensitive. So basically, again, the concept, coupling together a transducer, which is something that creates a measurable response, with some type of selective recognition component. And that's the basic concept behind a biosensor. So how can we achieve selective recognition if we're interested in selectivity? Well, we can't use off-the-shelf polymers like polyethylene or polypropylene that you would use for making some type of structural material. And these are called homopolymers because they could potentially interact with the polymer, but it'd be very weak. Instead, if you wanted to improve selectivity, you could utilize biopolymers. So biopolymers inherently have multiple types of functional groups that can interact with a molecule, and they can bind more strongly than just a conventional homopolymer. So in biological systems, they're really well known for being highly selective. So you can imagine antibodies present on your B cells, where the antibodies are geared specifically for recognizing different types of incoming foreign pathogens or antigens that your body's trying to defend against. You can imagine also our olfactory receptors in our nose being able to selectively recognize different smells, right? So the way these work is that you have multiple binding sites that interact with this molecule to be able to discern between different types of molecules. And this was done in our laboratory through the use of something called evolutionary screening. So to identify selective receptors in our laboratory, we go through this process called phage display or directed evolution. And what it boils down to is basically like a fishing expedition on a molecular scale. We take a library of viruses, and these are engineered viruses. These are non-harmful viruses for humans, but they do infect bacteria, so you don't have to worry about that. So these engineered viruses, we have 1 billion different viruses, and each virus on its tail has a slightly different receptor. So you have basically a library would be a collection of books. Here we have a collection of viruses, 1 billion different viruses, each one with a different receptor present. If we take that library and we wanted to identify a receptor in that library that can bind to a target that we're interested in, let's say it's a particular molecule that could be a hazardous biotoxin, let's say, or an explosive molecule. Let's say we want to make an explosive detection sensor. then we can identify that molecule and code it onto a bead. And that's kind of like our bait, so to speak, if you consider this with a fishing analogy. So we would take a bead, it could be a magnetic bead, coated with this target molecule that we want to find a receptor for, a receptor that selectively binds to that target. So we just take our library and we dunk in our magnetic bead that's coated with our target molecule. We let it simmer, we let it mix around, and effectively the ones that have the virus that have very strong binders or strong receptors for that target will stick to that bead. And then we just use a magnet to pull out that bead, and then we can wash away the non-binders. And we've captured the remaining virus that are very strong binders, have good receptors, so to speak, for our target. We then just grow more of those virus that have the good receptors. So that's the evolutionary part, right? It's the survival of the fittest, survival of the strongest binders. Those replicate, we replicate those in bacteria. We redo the same process using more difficult binding conditions. So we add detergents and things like that. And we do this 4 or 5 times until we identify an amino acid sequence, which is basically our receptor sequence, right? And so taken from this, we can utilize that as a receptor that can bind to our specific target. So the one example that we have here is an enzymatic biosensor for detecting abrin poisoning. So abrin is, if you don't know what abrin is, it's kind of like ricin. Ricin is, and abrin is actually the most lethal biotoxin currently known, and it happens through this molecule abrin. And this molecule abrin will inhibit ribosomes from carrying out their natural processes of making proteins. And abrin is actually found in these nice-looking little Jaworski peas. I guess they do look a little bit toxic, right? But yeah, they're very, very poisonous. But you can trace if you've been poisoned by this particular compound or by this particular biotoxin, if you find in the urine of somebody who's been poisoned the presence of a metabolite, L-abrine. So L-abrine is this molecule here. But unfortunately, being able to detect that molecule is really only demonstrated by the CDC and using fairly complex mass spectrometry equipment. So diagnosis of abrin poisoning isn't the easiest thing because at first it seems as if you have normal symptoms of diarrhea, fever, and it could be among 100 other types of possibilities of what could be the problem, right? No one's going to necessarily think that you have abrin poisoning. Unfortunately, the fatality rate the fatality rate is very high, and it can happen within 48 hours. So quick treatment is really necessary. So going back to that idea of early detection equals better treatment. So these Jaworski peas that you find here are more present in tropical environments, but luckily there's no widespread use of this abrin as a biotoxin as opposed to ricin, because ricin is cultivated for a more industrial scale. But hopefully that never is the case. But nonetheless, hopefully you'll be reassured that we have developed an enzymatic biosensor. So instead of having a huge piece of equipment, machinery that recognizes the presence of the L-abrin, we've used directed evolution of an enzyme to recognize L-abrine. Basically taking that gene, mutating it, and looking at cells that express that enzyme in the presence of a compound that makes them turn blue. So we basically screen for very blue color-producing bacteria, and those were the more active mutants that had higher specificity binding to this L-abrine. And we implemented that into a colorimetric detection platform. So basically using this compound TMB, when it gets oxidized in the presence of this enzyme and in the presence of the L-abrine, it converts that into a detectable signal that we see by a blue color. And when we were able to detect this down to 4 to 8 micromolar, which was quantitatively relevant for detecting abrin poisoning. So unfortunately, as you can see nowadays, people are scrambling to produce low-cost detection platforms for things like coronavirus. Usually projects like this don't usually get the full attention until it becomes something that's incredibly urgent, but you would feel reassured that we have something now that can be used for a low-cost detection of abrin poisoning, should it ever be needed. Hopefully not. So let me skip ahead here a bit. So now that we know that we can utilize evolutionary screening for identifying selective recognition elements, I want to introduce the concept then of what type of transduction platform we're going to use. So in our laboratory, we utilize a polymeric nanoparticle system that was developed at UC Berkeley while I was there as a PhD student. They developed this pentacosyldienoic acid-based nanoparticle. So you don't need to know all the details, but just know that this nanoparticle is initially a blue color, and if something binds on the surface, it turns into a red color. And so that happens basically by rearrangement of the side chains. So any type of side chain rearrangement will cause these to rotate, and that rotation makes this blue nanoparticle turn into a red nanoparticle, just because it has a different absorption spectra. These also go from a colorless to a fluorescent red color as well. So a benefit of using this type of platform is that we can easily integrate onto the surface our receptors that we discover through evolutionary screening, and it's simple to read, inexpensive, and rapid, right? You can just directly identify that this color change is happening. So an example of this is basically we can take our receptor that we identified, add it onto this molecule that I mentioned, combine it with another molecule that does not have the receptor, and then display it on the surface of this nanoparticle. And these form by self-assembly, which is nice. So we didn't have to go through any rigorous fabrication approach. Literally, you mix these two things in water or in a buffer, they self-assemble and form these nanoparticles by themselves. What's nice with that is we can easily identify different types of receptors, display them on the surface, and examine whether or not they display a sensitivity to a particular type of target analyte. So we've looked at identifying different types of lipids. We've looked at recognizing small molecules like explosives. We've even recognized the presence of water using these types of polymeric systems. In this case, we have stomata mapping, which you may not be familiar with, but basically plants, and I should say leaves, have tiny little sweat pores that they use for transpiration process, which is a vital part of photosynthesis and regulating water. And here you can see these little stomata, which are, again, like the sweat pores of a plant. You can see when they release water, we can identify this by a red fluorescence turn-on response. So in this case, we didn't utilize the self-assembled vesicle. Instead, we just sprayed it directly onto the surface of the leaf. So these polymeric nanoparticles are basically then a way in which we can imagine to be a sensor. And the sensor, given that it has a proper selective recognition element on the surface, could be used for disease detection if we could discern between two different, if we could discern that selective recognition element can bind to a specific biomarker that indicates that disease. So for a biosensor to be useful, it needs to be able to detect and discern between something that's a healthy cell, let's say, and a diseased cell. So the terminology for that is usually a biomarker, which is just some type of entity or element that's present. So a molecule, for instance, that's on the surface of one type of cell, the disease cell, but not on the surface of another type of cell, like a healthy cell. So using these types of polymeric systems, we can imagine them being able to target specifically to, let's say, a cancer cell if we incorporated receptors on the surface that could bind to that specific biomarker. And in one manifestation of this, you can imagine this as being an imaging probe. So here we have magnetic resonance contrast agents embedded onto the surface of this polymeric nanoparticle. So in our laboratory, we're really interested in exploring this for cancer research, specifically diagnostics geared toward follicular lymphoma B cell detection. And so what makes follicular lymphoma B cells quite interesting is that they have a unique sugar structure displayed on their surface. So this sugar structure is the biomarker that I just mentioned. So if you look at what follicular lymphomas are, they're a subset of non-Hodgkin's lymphomas, which are blood cancers. And blood cancers make up about 11% of all cancers. You've probably heard of follicular lymphoma or non-Hodgkin's lymphoma recently, or if you watch television, because you've probably seen commercials for litigation or attorneys for not for Roundup lawsuits, for instance, right? Because Roundup had been identified as a causative factor of non-Hodgkin's lymphoma. So, and over time, we've been seeing that non-Hodgkin's lymphoma cases have been on the rise. So follicular lymphomas in particular are an interesting subset of those because they start out as displaying this oligomannose or high mannose on the antibody presented by those B cells. So if you can discern by selectively recognizing this oligomannose structure or high mannose structure, then you can identify that follicular lymphoma subset. So can we identify a receptor for this oligomannose by evolutionary screening? Long story short, we tried multiple times and it didn't work. So evolutionary screening works, but just like any type of fishing expedition, sometimes you go fishing and you come back empty-handed, right? But that's how it goes, right? If we looked into the literature further, you could see that it wasn't necessarily an easy thing to fish for in the first place. People have been researching oligomannose binders for decades, actually, because oligomannose is also the same glycan present on the HIV virus. So researchers have been working for a long time trying to identify receptors that represent selective binders for this type of sugar. But serendipitously, an antibody was discovered in a patient that had HIV, and they identified an antibody that would bind to that oligomannose displayed by the HIV virus. This antibody was unlike any that people had seen before, in that it didn't have this typical Y shape that you would see for an antibody. Instead, it looked more like an I, right, rather than a Y. So a really interesting heavy domain crossover happened where you had a huge surface area available for binding to the oligomannose on that virus. And here's a structure of what that oligomannose looked like. So you have 1, 2, 3, 4, 5, 6, 7, 8, 9 mannose units. We also engineered a natural lectin found from bacteria. That's the Pseudomonas fluorescens agglutinin, PFA, I refer to it for short, because it was also known to bind to oligomannose. So we engineered a variant of this PFA and examined it as well. So we took this 2G12 and we saw that yes, it indeed does bind to the oligomannose displayed by the HIV particles, the HIV GP120 specifically. That's the glycoprotein coat that displays oligomannose Man9 units. And we developed a particle-based assay, which was agglutination-based or aggregation-based. So we coated the surface of these beads with our PFA, our lectin, which binds to oligomannose, and then we introduced it into the system that contained oligomannose. And we can identify that over time, 40 minutes to 80 minutes, we see aggregation occurring because this oligomannose basically serves as the cross-linking agent, causing agglutination of these beads. So it's important to note that if you're going to make a detection system, the most common are going to be color-changing systems or aggregation-based systems if you're going to be using a visual readout rather than an electrochemical or electromechanical system or an optical system. So, and again, in this case, the sensitivity is going to be limited specifically to how small of these particles you can see as an aggregate. So how many of these particles you can get to aggregate together to cause a larger clumping. And for the prior case that we talked about, like the case of the color changing from clear to blue, for the case of the L-abrin detection, that's going to be limited by how well you can see a color change. So it's important to understand that those sensitivity limits do exist when you're using a more widely deployable and inexpensive non-electrochemical system. So since this worked really well, we want to test it in the case of follicular lymphomas. So we ran into a roadblock in that there were no follicular lymphoma B cell lines available. And so we went through securing patient follicular lymphoma B cells. But the follicular lymphoma B cells, when you take them out of the patient, they don't grow anymore. And so that's why you don't have a set of cell lines available for follicular lymphoma B cells. So we decided to develop a mock follicular lymphoma cell line that still displays the oligomannosylated antibodies. So we isolated the DNA from a follicular lymphoma tissue section. We did next generation sequencing and identified the sequence of the antibodies that made those oligomannose displaying glycans presented on the surface of that B cell. And then we basically cloned those into a set of cells, which we refer to now as the BZ cell line. So they're not B cells, it's a mock B cell line. We call it the BZ cell line. And we tested these in mice and saw that they display antibodies and confirmed that they display the oligomannose. And then in the course of doing this experiment, we tested the 2G12, we tested the PFA. Neither of those worked perfectly in identifying non-discriminately the oligomannose. But we didn't give up. We looked to, and luckily, at the time, a publication had come out identifying that DC-SIGN and DC-SIGNR were identified to be potent binders of this oligomannose. And these are endogenous lectins that are displayed inside of our body on macrophage, and were actually the stimulating factor to cause activation of these follicular lymphoma B cells, or this malignancy, in follicular lymphoma patients. So this DC-SIGN and DC-SIGNR, we took this lectin and mobilized it onto our magnetic beads, and sure enough, we were able to discern between our mock follicular lymphoma cell line, the BZ cell line, and the background control cell lines. So DC-SIGN, and we also found DC-SIGNR to identify higher levels of enrichment, so we could actually capture and select these follicular lymphoma, mock follicular lymphoma cell line or cells versus the control cells. So now we have the selective molecular recognition element, and these are results that we haven't published yet, but they're coming out probably in the next month. So from here, we're going to move on to integrating this into our nanoparticle transducer. And once we incorporate that, we'll complete our testing of our sensor. So how we're going to go about creating this whole cell sensor, because now we're looking at identifying the presence of an entire cell rather than a small molecule. Again, this could be aggregation based, but first we need to identify whether or not our system works well with whole cells. And we looked at the prospect of this, this same polymeric PCDA amplifier that I mentioned before for generating these vesicles. So we used the pure PCDA vesicle itself. We made a modification of the PCDA to basically create a positively charged vesicle. So now you can, if you want to think about it simply, you just have this nanoparticle here that's white, which is negatively charged or uncharged. And you have this green nanoparticle that's either uncharged or positively charged. And so what we're doing here is really identifying how does the surface charge affect our level of non-specific binding. Because like we said before, we want to have a sensor that works really well in recognizing only this oligomannose. We don't want it to bind to everything, right? And so if you have a huge non-specific binding event, that's going to cause false positive signals, right? And we know that false positive signals and false negative signals are things that you don't want to have in a sensing system. So we found that when we increased the proportion of these, we could actually go from something that was relatively non-specific, or sorry, something that was had relatively low levels of non-specific binding to things that had very high levels of non-specific binding. And particularly found that when we increased the amount of PCA EDA content, that's made it more positive, these positively charged nanoparticles would interact more nonspecifically with the cell lines. And so we found this actually to be a pH and charge dependent event. So we looked at just the particles by themselves, no cells present. We could see that at low pH, those that were the pure PCDA nanoparticles actually aggregated by themselves with nothing added just because of the low pH event, because they went from being negatively charged to being neutral. And when there's no charge on the nanoparticle, they'll actually stick to each other. You need to have some amount of charge present to repel and basically maintain a stable colloidal solution. If you looked at the predominantly positively charged nanoparticles at very high pH, these would aggregate. But you can see that we could effectively tune this region to be stable and non-aggregative. However, the best was that of which we had a pure positively charged vesicle. Let me get back to this slide, but a problem with having that positively charged vesicle was that when we looked at the cytotoxicity, we found that those that were very positively charged, if you look at these in the second two bars of each of these representative columns, you see that you have a very low cell viability for the use of increasing amounts of these positively charged nanoparticles. So if you do use these nanoparticles, they would actually insert themselves into the cell and kill the cell. And that happens in a pH-dependent manner. So another interesting thing that we saw, if we just looked at the PCA, the original nanoparticle polymer formulation, and we compared BZ versus the hex cells, which are the background cells, we saw that, interestingly, we could already discern between these two types of cell lines. But this was non-specific discernment between the cell lines, and it's a result of the fact that the BZ mock follicular lymphoma cell line has this negatively charged antibody presented on the surface. So you actually have less interaction of that BZ cell line with the nanoparticles. So you have the prospect of a false negative signal in that case as well. So to overcome this, we effectively had to incorporate some form of stealth motif. And the most common one of these is polyethylene glycol. So polyethylene glycol is basically, like I like to refer to as a molecular windshield wiper. It just has a very water-soluble, highly polar composition that allows it to move around, and it doesn't want to be masked effectively. So incorporating this in a small percentage into the nanoparticles, we actually saw that we could dramatically decrease the level of nonspecific binding to the cell lines. So in our future formulation, we're going to take this nanoparticle that we've developed that has polyethylene glycol, and then we're going to display this DC sign that we mentioned before onto the surface and see how that fares. So taking that nanoparticle now that we've avoided nonspecific interactions with, we're going to expand this technique. So we have a nice platform. We want to expand it for antibody detection in general. And so to do that, imagine this concept of taking a sample of your blood. Let's say you wanted to detect the presence of, if you've been exposed to coronavirus or not, right? So you guys have probably heard of antibody testing already. So in this case, you could utilize the polymeric nanoparticle we just mentioned before, and you can incorporate something that represents the coronavirus, SARS-2 coronavirus. So for instance, it could be the spike protein. So you have the spike protein displayed on the surface of this polymeric nanoparticle, and if you have antibodies present, those nanoparticles will be bound by those antibodies, and they will aggregate and clump together inside of the test tube. If you do not have antibodies in your blood serum, then if you introduce nanoparticles that have the spike protein coated onto the surface, you will effectively have no clumping, and you'll have a negative signal. So we're interested in actually utilizing this for our future aim of looking at sugar-specific antibodies in the human immune repertoire. And again, this goes back to the concept of what we just mentioned before, of that certain cancers display interesting and unique sugars that you wouldn't normally see. And so if your body already has something that recognizes those sugars, it's very indicative that you may have a particular type of disease. These could be, depending on the sugar, IgA nephropathy, it could be Crohn's disease, or different types of cancers. So being able to screen and identify if your immune system is possessing these types of antibodies could be an indication that you have low levels of a disease already that haven't been manifested by larger scale or worse symptoms, but offers the potential then for utilizing that for early detection. So we're going to expand that technique, expand these nanoparticle systems in that direction. So a summary of that and our future efforts. So we have this nice modular platform for sensing in which we utilize molecular recognition elements embedded onto the surface of this modular nanoparticle system. We can tune the surface density of the molecular recognition elements that we incorporate onto the surface. We can incorporate different charges on the surface, and we can incorporate this stealth motif of the polyethylene glycol as well to minimize nonspecific binding. And in doing so, we can minimize false positive signals or false negative signals. And so we're going to continue these efforts. And here's basically a representation of what we'd like to do for our immune repertoire detection system. We would identify basically the molecular recognition element, that being in this case the sugar itself, and then identifying whether or not you have the presence of those antibodies by mixing your serum in a test tube with this nanoparticle system and look for either a change in color or, as we saw before, a change in the aggregation state. So I'd like to thank you all again for listening to my talk about really just the tip of the iceberg of what we're working on in our laboratory. There's lots of different projects that are available. I should say that the projects that I outlined today were done in a large part by help from undergraduate students. And so we have actually an NIH-sponsored R15 award for undergraduate research. If any undergraduates are listening out there and are interested in participating in research in our laboratory, please contact me. And yeah, I'd like to open it for any questions. Thanks.
Thank you, Justyn. That was a fascinating presentation of your work. So I think what we're going to do at this point is open up the, I guess, the chat window for questions. And anybody has any questions, please type it in the chat window, and I'll take the effort of reading it out. So we already have one question. So this is a general question. What are some of the networking opportunities in this field? So I'm trying to understand what networking would mean here. Justyn, over to you, if you think you can interpret this for us.
The networking opportunities? Well, if you're referring to how to get involved in it, then you would want to try to get integrated into these on-campus activities that we have through the Genetic Engineering Society and what's called iGEM. So Genetic Engineering Society and iGEM are student organization. Well, Genetic Engineering Society is a student organization at UTA that really sponsors the iGEM team. And basically, iGEM is this genetically engineered machines competition. So kind of like battle bots for genetic and biological systems. So you can build, design, build, and test different types of biological control systems. And you can implement those inside of a chassis, which would be either a mammalian cell or a biological cell. And then you can test those and basically try to make them produce some type of controlled output. So trying to engineer the biology in a better way. So these are like genetic circuits, so to speak. And so this is open to all engineers and non-engineers as well. In the past, we've had people from life sciences as well. So that's a nice networking opportunity in that respect. In terms of networking in another sense of the word, I don't know if I can answer that much because these would be non electrochemical or electromechanical systems. If you wanted a wide deployment of a sensor like this, you would have to couple that chemical or that color change or aggregation change into that visually discernible signal into some type of electrical signal, which shouldn't be hard because we use that all the time for quantification of it in our laboratory. So basically looking at the change in absorption spectra or a change in the transparency of a liquid.
Thank you. So all your work is mainly targeted towards detection of diseases, right? So which diseases, in your opinion, have been the most difficult to detect over the, I would say, last few decades, and where do you think your particular approach is going to be the most promising?
Well, I would say one of the most complex issues is there's a lot of heterogeneity in diseases that we're not aware of and we don't fully appreciate yet. Okay. So let's say, I don't know, 40 years ago, if you were told you had cancer, it wasn't as clear of what form of cancer you may have had. Nowadays, with all the types of molecular profiling techniques we have, we're much better at discerning what type of cancer one person may have versus another person. And effectively in that regard, each person may respond differently to treatment than another person because of those slight heterogeneities. Even in the case of the non-Hodgkin's lymphomas that I mentioned before, just recently people have noticed that certain subsets of these diffuse large B-cell lymphomas actually also carry this oligomannose sugar, and they behave differently and respond differently. So the detection of certain diseases really requires that one have a biomarker present to detect it early before it becomes a largely detectable symptom. And for the most part, clinicians are utilizing immunophenotyping techniques coupled with histology and just general examination, as well as genetic and molecular profiling to identify and discern between different types of diseases. But that's not to say that there aren't possibilities of us in the future refining those definitions into even better categories.
I see.
But yeah, it mostly relies on, there's like a few workhorses, so to speak, in the clinical pathology area. Those are flow cytometry, right? And you have immunohistochemical staining and something called FISH, like basically detecting genetic level variations. But they all work together along with how the clinician investigates the symptoms of the person, along with imaging modalities to really try to pinpoint what's wrong. So they all come together in a way. You can't really have, I shouldn't say that we can't have pure confirmation of a disease by one simple test usually.
So do you anticipate some of these detection techniques to become a part of annual physical exams and all that, or do you, or do you see that this will happen only when a patient comes to a doctor sick, and then some of these mechanisms are put into place?
Yeah, I presume as things move forward, people have started to really take an interest in their health on a personal level. So you see like 23andMe, people looking into their genetic traits, and people are nowadays trying to even more simply identify what genes they may have that could lead them to future diseases. It opens up a lot of interesting areas, and I think really the personalized and if you want to call it, I guess, home medicine is becoming more popular. So having a modular system that works very well is kind of critical if one were to have something that's cheaply deployable, let's say like a Walgreens, so to speak, if you wanted to identify if you have certain disease markers.
I see.
It'd be much cheaper than, I shouldn't say much cheaper, but more easy to do on your own than a genetic molecular profiling approach, which maybe in the next decade will get much cheaper.
Okay. So it's just a matter of cost right now?
Yeah.
Besides, okay, I see.
And there's also the differences is that some things you can detect at the genetic level, some things you can detect at the protein level, and then like I mentioned here, sugars. There's no genetic tie to sugars, so your DNA sequence may look perfect, but you might have odd sugars presented on your cells that normally wouldn't be there. In a way, that does tie to the DNA sequence because you have glycosylation sites, but you have billions of those or millions of those.
Yeah, it's pretty interesting how detection is such a complex molecular level, process. Is enough effort put into predicting diseases? I mean, I know that there is this current sort of very broad, ways of predicting whether you'll have diabetes or whatever, right? I mean, if your sugar levels are high, so they will predict that within 10 years you'll get diabetes and so and all that. So do you think there is a lot of research going on in maybe predicting future dangerous diseases? I know you mentioned based on your genetic profile, you may be able to do some predictions, but how good are predictions these days?
I think that's still like a really growing area. So a lot of ways in which people are trying to make these, how do I say, kind of algorithms, so to speak, for determining whether or not somebody is going to have a certain disease. Mm-hmm. I think right now there's still a lot of data being gathered, but as these, I don't know if you would call them trials or not, but as this clinical research gathers more and more data, I hope and expect that we would see that there's a much higher predictive value in looking at these panels, like these metabolic panels, for things that we didn't previously know about. So, so a lot of research is going on in the realm of looking at RNA expression levels and looking at even genetic level variations to see whether or not these, in a way, they are themselves biomarkers, right, can be indicative of certain diseases. So in our research, in the future, I'd actually like to take this forward into looking at if one could, so to speak, crowdsource the idea of immune repertoires, which are your antibodies and your T-cell receptors, basically the sequence of the different sequences present in your immune system, to look at all the patients who have done well versus those who have done poorly in responding to treatments or with certain diseases, and identifying if there are any commonalities or common sequence features that were indicative of better outcomes. So there might be some prognostic value in knowing whether or not you have this type of antibody present, as opposed to another type of antibody.
I see. Interesting.
So I think it's going to be a very growing area. Mm-hmm. It already is, I think.
Okay. I had a couple of specific questions about your research. Well, one is not that specific. How do you get the material needed, the lab material needed for conducting your research? How do you get, let's say, a patient who has a certain disease versus a patient who doesn't have a certain disease, so that you can measure false positives, false negatives, things like that?
Yeah, so for that, generally you will want to pair with the hematopathologist.
Okay.
but there are lots of resources available in which you can get de-identified samples, as long as you've gone through the proper channels. So it's good to have partnerships with the local hospitals. So, for instance, our laboratory, we communicate with the hematopathology lab at UT Southwestern, and they have a nice flow cytometry system set up there, as well as the one that we use here. And so, for basically doing different types of screening, they can do the preliminary clinical validation of which type of cancer, for instance, is present, and they can allow you to have those samples as opposed to those that are non-positive for, let's say, diffuse large B-cell lymphoma. So they make the call, so to speak, on if this patient has had or has currently a particular disease.
So does it mean that without these partnerships, it's going to be very hard to do research in isolation?
Yeah,
so there's no public resources where you can maybe purchase this through a grant acquisition?
Yeah, there are. Yeah, there are.
I see.
We developed our own cell line actually because there was no existing entity that, because if you had follicular lymphoma B cells and you try to grow them in vitro to basically amplify them or get more number of those for your testing, they'll die off in a few days. So unless you have something that's a dedicated cell line, then you're limited to doing the research in a very short window.
Can you shed some light? I know this may not be your line of work, but I mean your immediate line of work, but can you shed some light on COVID-19 detection research that's going on, where things are? I'm sure things are moving at a frenetic pace. But just where things are right now, how accurate they are, what's the false positives, how invasive are these?
The gold standard systems are the PCR-based systems, and those have done very well and are reliable. There's no sensor that's 100% perfect. So when you're talking about testing millions of people, I guess a 99% true positive rate or a 1% false positive, false negative rate could be kind of sizable. But the best part in what I hear a lot of testing that's being done now is they're doing simultaneous testing by multiple modalities, so that you don't have to worry about such a high error rate, because you have this cross validation, so to speak.
I see.
So the testing quality, I think, is good depending on what tests are being done, because I've heard that some of the tests that were provided to the state of Texas were passed over for the ones that we'd already had in-house because they were faster and more reliable. So
so when you said we had in-house, who do you mean?
Well, I mean the ones that, for instance, maybe it was back in April or March, or whenever the testing started, there's a lot of talk on the news about how certain tests were being deployed to the state of Texas. And then following up on that, the institution said that they preferred to use their own tests that they already had in place because they didn't require 2-week turnaround times, right? Because you could get results faster, and they were more reliable. So
your nanoparticle-based testing, what's turnaround time?
Yeah, some of those can be pretty long.
Oh,
so like 48 hours, but some of them can be fast. Yeah.
Okay, I see.
Yeah, PCR-based detection techniques are usually pretty rapid, but depending on the concentration of the nanoparticles and the concentration of the target, it can fluctuate. So we have a really high concentration of a particle and a very high concentration of a target. You can see a very bright signal in a very short time.
Okay.
But conversely, you might have to wait about 48 hours or 24 hours. Sometimes you can see it in as little as a few hours, like the examples I showed.
So Justyn, we have about 5 minutes left. Do you want to share with the audience anything else that you're doing that is interesting? Or maybe talk a little bit about how students can join your lab and things like that.
Yeah, so so in our laboratory, we recently received this NIH grant to support undergraduate research. So we already have a set of students who have been working on this project for the last year. They've been doing a really great job. They'll be graduating soon, so now's a good time to come and learn. I and the students could also train any undergraduates interested in joining, and I think they've had a really great experience. They've had already a publication and another one that's in preparation, and they've attended virtual conferences, I should say, much like this one. So it's a good chance to get to hone your skills and grow new skills. And ideally, we would like to see students who are in their junior level. As you get into your senior level, you become very busy with your senior design projects as engineering students. But junior and senior level students are welcomed. If you're interested in doing research and you're a freshman or a sophomore, that's a good place to join the Genetic Engineering Society and the iGEM team, because through the iGEM team, you can basically learn more about synthetic biology and techniques, including cloning and PCR-based techniques for cloning and subcloning techniques. You can learn about, in this case, our students develop microbial fuel cells. Basically, it gives you a good way to start getting your hands wet, so to speak, and doing lab research. So I'd like to invite students who are interested in that to look into the Genetic Engineering Society, because it's through that organization. Joining that organization is when you'll find out about these iGEM opportunities, which unfortunately this summer we couldn't have iGEM because of restrictions, but I anticipate next summer will be open.
Great. Well, we're just about closing in on the hour, so I'd like to take this opportunity to thank Dr. Jaworski for this fascinating discussion on his research. Sounds very topical, very interesting.
Thank you.
I hope everybody enjoyed this 1 hour that we all spent together. We will be continuing this virtual brown bag for the foreseeable future, so I look forward to welcoming everybody another time. Thanks, everybody, and have a great weekend.